Imagine typing cure for pulmonary fibrosis into a search bar. You hit enter, and instead of getting a list of Wikipedia articles or, medical journals, a fully autonomous system just boots up.
Right, exactly.
And then 12 to 18 months later, it spits out a clinically validated, FDA-ready drug candidate that has never existed in nature.
Never existed, yeah.
No human trial and error, no pipettes, just mathematics and code. I'm your host, and today we are opening up a massive, highly technical 2026 report titled The Architecture of Pharmaceutical Superintelligence to find out how this is rapidly shifting from, science fiction to our current reality.
And I'm thrilled to be here. It really is a profound shift in how we approach biology.
Mm-hmm.
Historically, the pharmaceutical industry has relied on this incredibly fragmented, human-led process of trial and error.
Right.
You find a target, you screen hundreds of thousands of random compounds, and you just hope one of them happens to stick.
You're just throwing darts at a board, basically.
Pretty much. And it's a process that pours billions of dollars and decades of research into a funnel, resulting in a staggering 90% failure rate in the clinical stages.
90%. I mean, if you were building airplanes and 90% of them crashed during testing, you would fundamentally change how you engineer airplanes.
Exactly. You'd have to.
And that is the mission of this deep dive today for you listening. We are exploring a fundamental phase shift. The industry is moving away from what the report calls assistive AI, which is basically just a digital assistant helping a human chemist organize data, to a fully autonomous prompt-to-novel therapeutic pipeline.
But the core of this transition relies on acknowledging a hard truth. The report freely admits that generative AI tools in their current state are imperfect.
Right. They hallucinate, they make mistakes.
They absolutely make biological mistakes today. The true magic of the ecosystem we're analyzing today isn't that it is flawless right out of the box. The magic is in how the architecture is built to self-learn.
Okay, let's unpack this. Because the report details a system designed to systematically eradicate its own flaws, avoid getting stuck in bad mathematical traps, and ground itself strictly in real-world physics.
Right. Relentlessly approaching 100% precision over time.
Right. And to understand how an AI can do all of that, we first have to look at how it actually reads human biology. Because historically, proteins, which are, the little molecular machines running your body, they've been incredibly messy for computers to comprehend.
Oh, massively. The primary struggle has always been that protein backbones are continuous. They're these wriggling strings of amino acids floating in three-dimensional space.
Like tangled noodles.
Yeah, exactly. And if you simply translate the atomic coordinates of a protein into a raw spreadsheet of numbers, the computer loses a massive amount of critical functional context.
It just looks like noise.
Right. So the AI's very first job, if it actually wants to fix that 90% attrition rate, is to understand 3D protein structures with absolute perfect clarity.
And the report introduces a mechanism for this called geometric byte pair encoding, or GOBPE. I was trying to wrap my head around this because it sounds incredibly dense, but it reminded me of how we process text.
Oh, that's a great connection.
Yeah, because if you think about how a large language model reads an article, it doesn't read whole words, right? It chops words down into discrete tokens or syllables.
Precise.
It sounds like this system is doing the exact same thing, but instead of text, it's tokenizing 3D geometry.
That is a highly accurate way to visualize it.
Mm-hmm.
By utilizing GOBPE alongside a protein structure tokenizer, or PST, the system takes continuous spatial curves and transforms them into discrete sentences of geometry.
Wow.
It establishes a linguistic blueprint for biology.
Think about it like this, for everyone listening. Imagine you have a messy, continuous clay sculpture of a protein. Standard computer looks at that clay and just sees an overwhelming, unreadable blob of data points.
A total mess, yeah.
But this geometric tokenization takes that continuous clay sculpture and breaks it down into a perfectly categorized box of discrete Lego blocks. Suddenly, the AI can read the physical shape of the protein the exact same way it reads a sentence in English.
And once you establish that linguistic foundation, the ecosystem deploys foundational models to actually begin the design phase.
Right.
The report highlights a model called Pearl, which handles protein ligand co-folding. The ligand being, the potential drug molecule that we want to bind to the protein.
Right, E fitting into the lock.
Exactly. And Pearl incorporates a diffusion module that inherently respects three-dimensional rotational symmetries.
I noticed that specific detail about rotational symmetry. It essentially means that no matter how you rotate the protein in virtual space, the AI understands it's the exact same object.
Right.
It doesn't get confused if the molecule is just upside down.
Which drastically reduces the amount of computational power and training data required. By combining real experimental data with massive amounts of synthetic data, this model achieved a 14.5% improvement over older models.
Like AlphaFold 3.
Yeah, exactly, over AlphaFold 3. And that's in generating physically valid, highly accurate molecular poses. It proves that the AI can place every single atom in the correct location.
But translating biology into a static language, it feels like it introduces a massive blind spot to me.
How so?
Well, grammar rules are great for writing a beautiful sentence on a page, but biology isn't sitting still. If we just teach the AI to read a frozen 3D snapshot, don't we run the risk of the AI designing a drug that perfectly fits a frozen protein, but completely fails the moment that protein starts moving in a human body?
What's fascinating here is that the architects of this system view that exact limitation as the dividing line between assistive tools and true superintelligence.
Oh, really?
Yeah. A static snapshot is completely inadequate because a protein is a dynamic machine. It breathes, it shifts, it alters its shape depending on its environment.
Right.
Therefore, the language the AI learns must capture temporal dynamics.
And the report details a tool called Stripes that addresses this, right? By representing protein interactions as symbolic sequences derived over time.
Think of it as transitioning from photography to cinematography. Instead of looking at a single frame of a drug fitting into a protein pocket, the AI is reading the entire sequence of events.
Watching the whole movie.
Exactly. How they approach each other, how the water molecules are displaced, and how the chemical bonds lock together over milliseconds. It achieves a dynamic understanding of biological recognition.
Knowing how the movie plays out is a massive leap forward. But when an AI learns by watching a movie, it naturally looks for shortcuts to solve the problem faster.
They always do.
It might find a chemical design that looks, good enough to fit the protein and simply stop looking for the actual optimal cure. In machine learning, this trap is called a local minimum, right?
It is one of the most dangerous mathematical traps in artificial intelligence.
Yeah.
Imagine you were trying to find the lowest valley in a massive, foggy mountain range. You walk downhill until you hit a dip, and you stop, assuming you've reached the absolute bottom.
Because you can't see anything else through the fog.
Exactly. But you don't realize you are just in a small crater near the peak. The true valley is still miles below you. That is a local minimum.
Wow.
The AI thinks it has found the ultimate molecular design because it looks better than all the immediate nearby options.
So the system needs a way to realize it's trapped in a crater and essentially rewrite its own search parameters to escape the fog.
Precisely.
This is where the report introduces the Darwin-Goodall machine, or DGM. This concept is just wild. DGM allows the AI agents to autonomously rewrite their own source code.
It's incredible.
I mean, think about the smartphone in your pocket. When it updates, it just downloads code that a human software engineer wrote. This system is acting as its own software engineer.
The agents are continuously modifying their own workflows, searching for better algorithms to explore that foggy mountain range. But the crucial element is how they validate those self-made modifications.
Right. How do they know it's a good update?
They test every new piece of code against rigorous scientific benchmarks, like SWE bench. If the new code successfully solves a complex biological puzzle better than the old code, the system integrates it.
Oh, wow.
It gathers these successful code mutations as stepping stones along a path of endless innovation.
And it orchestrates this self-learning through a subsystem called Agent Evolver. It uses something called self-questioning to automatically generate its own training tasks and self-attributing to hand out precise rewards.
Exactly.
So if the AI changes one single carbon atom on a massive drug molecule and the binding affinity suddenly improves, the system knows exactly which specific edit deserves the credit.
And we can observe the immediate impact of this through a mechanism called test time training, or protein TT.
Okay, what is that?
Traditionally, you train an AI model, you freeze its neural weights, and you deploy it. It relies entirely on what it learned in the past.
Right, it's a finished product.
But test time training allows the model to customize its own understanding of a single, highly specific target protein right at the moment it is trying to solve it.
I was actually reading this section on test time training, and I kept tripping over this word, perplexity. The report states that the AI improves its accuracy by minimizing its perplexity when it encounters a unique protein pattern.
Yeah.
How does an algorithm actually experience surprise?
It's a brilliant way to frame statistical uncertainty. When the AI encounters a novel viral protein, its initial predictions will have a high degree of variance. It is statistically perplexed.
It's confused, basically.
Right. By running continuous simulations and minimizing that mathematical uncertainty on the fly, it actively adapts its internal representations.
I see.
On challenging viral targets, this localized self-correction improves structural accuracy by up to 19% without requiring a complete retraining of the model.
Okay, here's where my alarm bells start ringing.
Oh.
If an AI is rewriting its own code, generating its own training tasks, and handing out its own rewards to minimize its own surprise, how do we know it doesn't just learn to cheat the test? How does it avoid getting stuck in an endless loop where it's just generating terrible chemical designs but giving itself high scores because it wrote the grading rubric?
That is the defining vulnerability of any closed-loop AI system. If it only references itself, it'll eventually drift into delusion.
Right.
To combat this, the ecosystem is structurally forced to interact with empirical biological environments, utilizing a framework called Skill Foundry.
So Skill Foundry acts as an anchor to human reality. It synthesizes validated procedural knowledge from actual real-world research papers and code bases.
Exactly. The AI self-evolution is strictly policed by external empirical benchmarks. It cannot simply rely on its own internal logic to declare a molecule successful.
That makes sense.
It has to pull verified skill packages from actual laboratory data, ensuring its self-generated rewards align with physical reality.
Which brings us to the ultimate reality check. The literal laws of physics. Because code can be flawless, but if the self-evolving intelligence starts designing fantasy molecules...
Right, things that can't exist.
Yeah, structures that look gorgeous on a computer screen but immediately explode or fall apart the second you try to synthesize them in a real test tube, the entire pipeline is useless.
Anchoring the system to thermodynamics requires overcoming what researchers call the timescale problem.
The timescale problem.
Yes. Biological transitions, such as a protein folding or a drug slipping into a binding pocket, occur over milliseconds. But classical molecular dynamics simulations must calculate movement in femtoseconds to capture atomic vibrations.
And a femtosecond is what? 1/billionth of a second?
Something like that, yes. Unfathomably tiny.
It is an unfathomably tiny slice of time. Trying to simulate a whole millisecond by calculating every single femtosecond is like trying to watch a flower bloom by taking a million photos a second of a single petal.
That's a great way to put it.
You completely miss the big picture. By the time you process the data, you've missed the entire event.
And you have burned through an unjustifiable amount of computational power. That barrier is broken by something called deep jump. It's a generative model that provides up to a 1,000,000-fold acceleration. That's 10 to the 5th or 10 to the 6th power over classical simulations.
Wow. So, following the flower analogy, deep jump acts like a smart time lapse. It completely skips calculating all those microscopic vibrations and directly calculates the grand transitions.
Exactly. It predicts the temporal evolution of the protein state directly, ignoring the high-frequency atomic noise. Crucially, it maintains strict physical integrity by using multiscale neural network potentials.
Okay.
The report notes it delivers the gold standard of quantum-level accuracy, which is called density functional theory, or DFT. But it achieves it with sublinear scaling on a single GPU.
Wait, on a single GPU?
Yes. It is executing impossibly heavy mathematics at unprecedented speeds.
And the reason it has to execute that heavy math is to secure the thermodynamic anchors. The ecosystem relies on vast databases, like the PPABI database, which has 329 protein-peptide complexes.
Right, with precise isothermal titration calorimetry measurements.
Yeah, the ITC measurements. Providing precise data for Gibbs, free energy, enthalpy, and entropy. I want to linger on this for a second because the report emphasizes entropy heavily. Why is it so critical for a drug-designing AI to deeply calculate entropy?
If we connect this to the bigger picture, consider what happens when a drug binds to a protein. You are taking two molecules that were previously moving freely and chaotically in water, and you are forcing them to lock together in a highly structured way.
Right.
Nature fundamentally resists that order. There's a massive entropic penalty for binding.
It's like trying to force two opposing magnets together while underwater. The environment wants them to drift apart.
Precisely. By forcing the AI to optimize for Gibbs free energy, often denoted as delta G, you demand that it mathematically proves the drug has enough binding energy or enthalpy to overcome that entropic penalty.
Wow.
It cannot simply draw a shape that visually fits into a gap. It must calculate the brutal thermodynamic struggle of forcing those molecules together and prove that the drug will physically stick to the target.
But knowing the choreography and the physical constraints still leaves us with a terrifying blind spot. Large language models, by their very nature, have a dangerous tendency to invent impossible realities.
Hallucinations.
Exactly. In a customer service chatbot, a hallucination is a fabricated refund policy. In pharmaceutical superintelligence, a hallucination is a toxic drug that causes liver failure.
It's a huge risk. The generative models utilized to discover novel drugs naturally explore massive, uncharted, continuous latent spaces. To find a cure that no human has ever conceptualized, the AI must be allowed to guess in the dark spaces between known chemistry.
Right.
But novelty inherently borders on hallucination.
So what does this all mean for safety? How does the system police its own imagination? If the AI knows the physics, why would it hallucinate an invalid or toxic chemical structure in the first place?
Because it is continually searching for shortcuts to satisfy its mathematical objectives. That is why the architecture employs the critic framework. It functions as the system's immune system against mathematical hallucinations.
So critic acts as a heavily armed immune system, but how does an immune system actually interrogate a mathematical hallucination before a drug is physically made?
It applies a relentless verify then correct loop. The moment the generative model proposes a novel molecular structure, critic forces it to interact with external code interpreters and search engines.
Oh, wow.
It utilizes an interrogation technique called matched molecular pairs, tracked by a workflow called WISP. It presents the AI with two molecules that are nearly identical, save for one altered atom, and demands the AI predict how that single change impacts toxicity.
It's essentially a high-stakes game of spot the difference. If the AI can't accurately explain how changing one carbon atom flips the molecule from safe to toxic, critic knows the AI doesn't actually understand the underlying chemistry.
Exactly.
It catches the AI generating numerical noise that merely looks plausible.
And this rigorous interrogation extends to comprehensive toxicity prediction. Specialized modules within the ecosystem, like Tox Predictor and DeepMetab, predict liver, heart, and kidney injury by identifying precise sites of metabolism long before any physical synthesis occurs.
That's amazing.
And every single decision regarding toxicity is automatically tracked and logged by ToxMCP.
Yeah, the report mentions ToxMCP creates a fully automated, auditable provenance track that complies with LSEOA plus principles. It generates an unalterable paper trail for every choice the AI makes, bringing everything up to regulatory-grade rigor.
Yes.
And we know this verify then correct rigor actually translates to the real world because of modules like AutoIND that automate FDA applications. The AI can draft complex regulatory documents, reducing writing time by 97%.
Incredible efficiency.
Yeah, dropping a 100-hour task to just 3 hours with zero critical errors. That proves the AI can be trusted with compliance.
When you combine all of these architectural pillars, the strict grounding in thermodynamics, the eradication of hallucinations, and the capacity for autonomous self-evolution, the pipeline achieves its ultimate goal.
Pharmaceutical superintelligence.
Exactly. It transitions into true pharmaceutical superintelligence through a paradigm known as latent reasoning.
This brings us to the hierarchical reasoning model, or HRM. This concept completely upends how we traditionally think about scaling AI. It decouples the amount of time the computer spends calculating from the actual size of the neural network.
Right.
It features a high-level module operating in a high-dimensional subspace to figure out the abstract, long-horizon scientific plan, while a low-level module rapidly executes the immediate steps.
It mirrors the organizational principles of the human brain. But the true breakthrough is inference time scaling. Historically, if you wanted an AI to solve a harder problem, you had to build a larger neural network and train it on vastly more data.
Wait, so instead of building a larger, more data-hungry neural network, HRM mimics the human brain by just taking a longer time to think in its hidden state before it gives an answer.
Exactly.
It literally just increases the thinking time parameter, what they call Mmax at inference, when faced with an impossibly complex scientific task.
The math bears that out, and this raises an important question about how we define intelligence. The AI is no longer a simple input-output tool where you hit enter and receive an immediate reflexive response.
It's actually pondering.
It engages in latent reasoning, it simulates hypotheses, evaluates potential failures, discards toxic pathways, and refines its strategy before it ever presents an answer.
And the real-world proof of this latent reasoning is already arriving. The report highlights an industrial milestone from early 2026, rentosertib.
Yes, rentosertib.
It was the first end-to-end AI-powered drug, and it demonstrated a 98.4 milliliter lung capacity gain in phase 2 trials for pulmonary fibrosis.
By the time the AI outputs a viable clinical candidate like rentosertib, it has orchestrated the entire scientific method autonomously. It has used simulated virtual models in silico twins to map the patient's biological response.
Wow.
It has optimized the chemical synthesis pathways using separoagents, and it has audited its own safety margins to regulatory standards with ToxMCP.
So to summarize the journey we've taken today, this 12 to 18-month prompt-to-drug pipeline isn't magic.
Not at all.
It is a highly disciplined, brute force combination of technologies. It relies on self-evolving algorithms like DGM rewriting their own code. It demands strict thermodynamic anchors like deep jump and PEPBI to prevent physical impossibilities.
Yes.
And it deploys relentless hallucination checks through Critic to ensure biological safety. For you listening, the overarching takeaway is that the future of medicine is no longer bound by the slow, expensive process of human trial and error. We are entering an era of setting the parameters and letting a highly disciplined, self-correcting intelligence solve the puzzle for us.
But exploring this architecture leaves us with a final thought to ponder.
Yeah.
If a pharmaceutical superintelligence can autonomously hypothesize a biological target, design the molecule, simulate the thermodynamic physics, test it on a virtual human twin, and write the FDA application entirely within a self-contained digital loop,
Yeah.
does the definition of biological discovery permanently shift from the physical laboratory to the silicon chip? And if so, what exactly becomes the role of the human scientist?
It is a paradigm shift that redefines human ingenuity. Thank you for joining us on this deep dive into the architecture of tomorrow's medicine. We'll see you next time.